Hello, thank you guys for coming. I'm actually really excited to be in the field today, in the field of medicine. And one of the reasons that I find this space so interesting is that we're really in the process of changing the way the world looks at medicine. I mean, if you think about most of human history, we've treated diseases with plants and prayers. And we're really moving into a period now, a new era, where we're beginning to treat diseases with cells, living cells, and information. And I realize that most of the field doesn't quite think about it in these terms yet, but what we're really starting to do now is program the body to make its own treatments. And my background is a little bit different than most, and I came out of computer science and physics. And what got me fascinated by this space actually was something we take for granted a lot, vaccines. And if you get a vaccine, suddenly a disease that used to kill you won't even give you the sniffles. And this seemed curious because nothing's fundamentally changed about the pathogen or your immune system. But it's just information. And so as a kind of computer scientist mentality, I got thinking, well, where's the hard drive and where's the file? And, you know, if we are based on information at all, I mean, think about it, without the information in your cells, we're a few bucks worth of chemicals sitting on the floor. With information, we're life. And if this is the case, why aren't we treating diseases with information? And I think, you know, the obvious reason historically is we didn't have access to it. You know, cells don't have USB ports. But I'm going to start this short presentation by asking you to consider what would the world look like, what would our field look like if we had an app store for the human body. And so here at Immusoft, we've basically developed a tool to produce long-lived plasma cells that secrete therapeutic proteins. So instead of making a treatment and injecting it, we take your cells out, program them, and put them back. And we think this opens the door to treatments that are wildly impractical today, because things like the half-life of the molecule, the stability of the protein, and how difficult it is to produce or purify don't apply anymore. And a perfectly awful example of why we want to do this and why this is an improvement is our first clinical indication, MPS I. So this is a rare genetic disease, a lysosomal storage disorder, in which patients can't produce an enzyme they need to break down waste sugars in the lysosomes of their cells. And it's catastrophic and lethal within about 10 years if it's not treated. And typically, these patients will either undergo a hematopoietic stem cell transplant before they're about 2.5 years old, or they go under enzyme replacement therapy. So they produce this enzyme recombinantly and inject it. But the enzyme is incredibly expensive. It costs between $250,000 and $1 million a year per patient. And it only slows the decline. And so we think that we have a better way of dealing with this. A lot of the problems with producing short half-life biologics and injecting them are really what happens in your plasma. So in the case of MPS I, the patients will go in once a week for a 3-hour-long infusion, and they get this really high level of it, but basically by the time they get home, the treatment's already worn off. And so by producing something like this 24/7, you radically alter the pharmacokinetics of it. So you allow this to absorb in tissues where it wouldn't normally come. And so the process is simple enough. We take the cells out, program them, and put them back. But the details obviously a little more complicated. And so I'll go into this a little bit in the time I have today. So a patient would come in for a blood draw or an apheresis at a local clinic, and the cells will be shipped to us, and we will isolate the B cells, just normal CD19 immunomagnetic bead selection. And then we can electroporate a non-viral vector into them to produce a therapeutic protein. So in this case, we're using the Sleeping Beauty transposon. And the real beauty of this is that it's vastly less expensive than the viral-based therapies. So our system, when we first started it, actually used a measles virus pseudotyped lenti, and then later a VSVG lenti. And we had budgeted around $1.2 million for virus alone in our phase 1. And with this, it's basically just the cost of DNA. So I think if you want to be able to use these kinds of cell therapies to not just treat a handful of patients, but to be able to treat hundreds of thousands, you really need to have a much more scalable platform. And so this has allowed us to do away with what was the most expensive part. And so in this case, we've modified these cells to secrete IDUA, and we can make a lot of it. And we can watch how much they're producing before we ever have to re-inject the cells. So we take these cells and then we expand them ex vivo. We've managed to push the expansion up to as high as 10,000-fold. So we can take into account a tremendous amount of variability in patient to patient. Typically, we only need to expand them on the order of 50 to 100-fold. But we've managed to optimize the process so that we can accommodate variance in patient and still reliably release a product. Once we have these cells expanding, we begin to mature them into plasmablasts and plasma cells. And so the cell that we're producing eventually is the long-lived plasma cell. And this is why, actually, if you were to take blood from an 80-year-old, you can still detect antibodies against smallpox. It goes back to the original vaccination thing that got us started on this. The cells that are secreting those antibodies were actually generated all those years ago when they got exposed to the vaccine or the virus. The half-life of the cells in vivo is about 17 years. But a short-lived plasma cell will die in a couple of weeks. And so the culture system is optimized to produce these migratory plasmablasts. They have a chemokine homing receptor that will guide them to survival niches where they can hopefully engraft. So the initial version of this technology came out of the lab of David Baltimore at Caltech. And the first iteration had used hematopoietic stem cells, and then they'd be cultured into plasma cells. And that process took a long time. So when we licensed this, we modified it to work with just the quiescent cells out of whole blood. And that allowed us to drop the time required to do this to about 7 days. So once the cells are matured, we can assay them for migratory ability and survival, and then inject them back in. And so far, we've only been able to put this into mice. We don't have our IND yet. But a single injection of these can produce more than 30-fold wild-type levels of IDUA. So we can vastly exceed the levels that would be necessary to be curative. I mean, one of the nice attributes of this first indication is that if we can get to about 10% of the endogenous levels, we can ameliorate most of the symptoms. But if we produce vastly more, there's not any toxic side effects from it. So we've actually pushed this. I think the highest we've managed to get from a single injection is over 200-fold endogenous levels. So the plasma cell is an incredibly powerful secretory cell. And in the mice, we can't get them to survive 17 years, obviously. The niche is not fully compatible with the human cells. But we can definitely track them out for months. And we've actually managed to increase the immunoglobulin levels considerably in this as well. So this is just a quick high-level overview of how the system works. We'll isolate the cells, culture them all in a central location, and then we freeze them down for shipment. And one of the benefits of this approach is it allows us to actually administer multiple doses from a single production run. And so the goal of this is that the cells, once they're frozen, can be thawed with minimal manipulation. And so you have the ability to increase the dose or to do multi-dose later. And I think this is a really a kind of a key benefit of this, in that unlike directly injecting a virus, you don't have any immune reaction to it. So if you would like to increase the dose later, or if it does wear off, you can just re-administer more cells. It also means you can use the same platform to treat multiple indications in the same patient. So far, we've managed to basically do everything except for the human trials. We can reliably produce the cells. We've been transferring the protocols into GMP, and have just gotten final guidance from FDA to design our GLP tox study. So our goal is to complete this and be in humans sometime next year. So we're doing the first study in MPS I as an investigator-sponsored IND at the University of Minnesota. It's the largest center for the marrow transplants for this disease in the country. And so we think we have a clear path into the clinic with this, and can build a good basis for using the platform in other indications. think one of the real beauties of this platform is that if you want to make a new indication or go after a new indication, it's as simple as changing the plasmid, since nothing changes about the manufacturing process. And so we have about half a dozen active collaborations now where people will send us a construct and say, hey, can you make this? And we can load it into the cells and send them back cells and supernatant and say, yeah, you tell us if it works, we can give you a license. And so we've tried to keep as open of an approach with this as possible, kind of under the belief that it'd be very difficult to commercialize every aspect of this ourselves. And so we really wanted to take a first indication into the clinic and then license it out to people who need to produce it. So the latest one that's not on here is a collaboration we're doing with Stanford, where we've made Pro-Collagen 7 for epidermolysis bullosa. And I mean, it just kind of highlights the breadth of this cell type. You wouldn't expect that an immune cell can make collagen, but it does, and it does quite well. And so this is a similar thing where they had a need to produce it and sent us the construct, and we loaded it in for them. So I think I'll highlight we're starting with these small indications, and the goal is to move to larger ones as we go, and we gain the ability to scale up production and greater tolerance from the FDA for this kind of approach. And so we had done the initial work actually making broadly neutralizing HIV antibodies and had reverted back to the rare disease for the first indication. But I think where we're really going with this is being able to treat much larger indications. And one of my favorites here is the age-related sarcopenia. We've started making a molecule called follistatin, which is actually inhibits myostatin. So myostatin is a negative regulator of muscle growth, and follistatin is what you make when you exercise. And so it gives us the ability to recreate the biochemical pathway of exercise when you're sitting around. I guess the American dream, get strong watching TV. But I think there's a much more valuable thing to be done with it, and that's treating muscle wasting with old age. I mean, you think about in the U.S., I think we're going to have 50 million people over the age of 65 by 2020, and not many people are really appreciating what that's going to do to the healthcare system. Physical strength counts for a lot. I mean, if I fall downstairs, I curse. If my grandma does, she goes to the ER or dies. And so making people strong, I think, will greatly improve their quality of life and mobility and reduce the healthcare cost. So I think I'll wrap it up here. Oh, I guess one more. Sorry, added one more slide. We've raised about $6.5 million to date, and we're actually closing an A3 right now for I think now it's a little over $3 million, and that should close by the end of next week. And we'll be out raising a Series B early next year to pay for the clinical trials. So if anyone's interested in that or has any questions, feel free to harass me afterwards. And I'll wrap it up here. If anyone has any questions, I think I have another minute or two. All right. Thank you very much.